Hi everyone. Today we're going to talk about Lab 12, which is using the Seahorse instrument to monitor LDH inhibition. So at this point, you should have already watched the 3 short Agilent videos about the Seahorse, and I just want to tie in the basics of the Seahorse to what we're interested in learning about LDH. So this whole semester we've been studying lactate dehydrogenase, and you may wonder why it matters, and why we can use Seahorse, which monitors metabolic flux, to look at LDH and LDH inhibition. And so to do that, we have to go back to an overview of metabolism, which hopefully at this point, most of you have seen metabolism in biochemistry, but if you haven't gotten there, then we'll go over metabolism again. So metabolism begins at glycolysis, where a glucose molecule is split up into 2-pyruvate, and in that process, it consumes 2 NAD plus and 2 ADP, and produces 2 ATP and then 2 NADH. And so ATP and NADH are high energy molecules that can be used as energy sources elsewhere in the body. And then pyruvate can be broken down further to produce more energy sources. And there are 3 possibilities, or 3 fates for pyruvate. We are not too concerned with anaerobic alcoholic fermentation. I think that's something you'd see more in yeast. But there are 2 distinct possibilities in humans. And so the first is anaerobic homolactic fermentation. So this would be in a situation where your body or your cells aren't able to get oxygen. In this case, this is where LDH is so important, because LDH takes the 2-pyruvate and turns it into lactate. So that's the reverse of the reaction that we see studied in this lab. And in the process, it takes the 2 NADH that are produced during glycolysis and oxidizes the NADH molecules to NAD+, which can then be shuttled back into more rounds of glycolysis. So again, this is when there isn't any oxygen present. So an example is when you're exercising and oxygen can't get to your muscle tissues. In that case, your muscles will switch over to glycolysis to burn through the glucose more quickly, although it's not as energy efficient. And the resulting lactate or lactic acid is what causes your muscles to burn, because acid is acidic, and acidic things, if they're high enough concentration, can burn. Now, under normal conditions, so normal meaning you have a normal amount of oxygen present, or that oxygen is able to get to the cells, you would go through aerobic oxidation, so through the citric acid cycle, and then oxidative phosphorylation. And like with anaerobic fermentation, oxidative phosphorylation takes those 2 NADH molecules produced in glycolysis and oxidizes them. It also produces CO2 and water. You can see there the water is consumed or the oxygen is consumed during oxidative phosphorylation, but it also produces 26 ATP molecules. So when you go through the glycolysis and terminate in LDH, or terminate in lactate formation, so glycolysis alone, you get 2 ATP molecules. However, when you go through all the way through oxidative phosphorylation, you get 26 plus 2 equals 28 ATP molecules. Now, one of the things, though, that's interesting, and why this occurs isn't necessarily well understood, but in cancer cells undergo oxidative phosphorylation, but they also undergo anaerobic homolactic fermentation, but they do it when oxygen is present. All right, so even though these cancer cells, in theory, are able to access oxygen, they still go through glycolysis, terminating with LDH conversion of pyruvate to lactate, and they don't always go through the full cycle through oxidative phosphorylation. And so cancer cells have a tendency to consume a lot of glucose. And so when you can feed a cell glucose, or this is also a case where if the cell has a high amount of glucose, it might end in LDH, the LDH reaction, and not go through TCA and oxidative phosphorylation because it wants to get rid of that glucose quickly. But cancer cells will do this anyway, even with oxygen. So they consume lots of glucose. And so you might hear doctors or nurses or medical professionals talking about how cancer likes sugar, while cancer likes breaking down using glucose and shuttling it through glycolysis. So if we take just another, perhaps a more simplistic look at monitoring oxidative glycolysis and OXPHOS, specifically to how we can measure those two processes in Seahorse, is if we have a schematic here, this is a cell and then a mitochondrion and then a zoom in of a blood vessel outside of the cell. So again, you can see here that to undergo glycolysis, which is going on in the cytoplasm, glucose must be taken up, and the first step of this process, converting from glucose to glucose 6-phosphate, is catalyzed by hexokinase. This in turn produces pyruvate. Pyruvate is converted to lactate by LDH, and then lactate is shuttled out of the cell. Now, one thing that was really confusing to me about the Seahorse when I first was reading up on it this past year is how can you monitor glycolysis by watching the proton production after you've corrected for other sources of extracellular acidification, such as the production of carbonic acid from oxidative phosphorylation. And that's because, the reason why I include this figure is because this figure makes it pretty clear. And that's because whenever lactate is exported from the cell through the monocarboxylate transporter, a proton must also simultaneously be exported out of the cell along with the lactate. So this is, I don't know if it's necessarily, be careful with the term using coupled, but whenever a lactate is transported out of a cell, a proton is also transported out of the cell. So for every mole of lactate that's exported out, you see a mole of protons. And so that's how the extracellular acidification can be used as a measure for lactate production. Now, the connection between oxygen consumption and oxidative phosphorylation, on the other hand, is a little easier to see because in order to go through oxidative phosphorylation in the mitochondria, that oxygen must be uptaken there. So again, what is the Seahorse actually measuring? With oxygen consumption, or the OCR is the oxygen consumption rate. And so we're measuring the rate, we're measuring how much oxidative phosphorylation, or at least how much glycolysis that terminates in oxidative phosphorylation is occurring, whereas ECAR is the extracellular acidification rate. And again, it measures LDH terminated glycolysis. In the case of the cells that we'll be monitoring this lab, aerobic glycolysis. And from now on and throughout this lab, if we talk about oxidative phosphorylation, we're making the assumption that it's glycolysis that ends in OXPHOS, so the whole metabolic cycle. But if we talk about glycolysis and glycolysis only, that's referring to glycolysis that ends with lactate production, and that's aerobic glycolysis, or glycolysis that ends in LDH, the LDH step, even though there is oxygen present. So now that we've gotten that out of the way, what are the questions that we are trying to answer with this lab? And so the questions that we answer is we're looking at two inhibitors. So if we zoom out a little bit here, we're looking at actually three different glycolysis inhibitors. So glucose is not an inhibitor. We're looking at 2-deoxyglucose, which is a modified glucose. So it's a glucose with the alcohol group missing at the 2 position. And then sodium oxamate. Oh, so that's 2-DG. Looking at 2 inhibitors, 2-DG, and then sodium oxamate. So in the kinetics module, we used oxalate as an inhibitor, but for this lab, we're going to be looking at oxamate. And whereas oxalate was a competitive inhibitor of the lactate to pyruvate reaction, oxamate is a competitive inhibitor of the pyruvate to lactate reaction. All right, so we're looking at these 2 different inhibitors, and they inhibit 2 different steps of glycolysis. So we know that oxamate is an LDH inhibitor, and 2-DG is a hexokinase inhibitor. So if you recall from the figure on the previous page, that's the first step of glycolysis. And so LDH is a competitive, oxamate's a competitive inhibitor of pyruvate. Hexokinase is a competitive inhibitor of glucose. So we're looking at the first step of glycolysis and the last step of aerobic glycolysis. So the question we want to ask, the questions we're interested in understanding, is how do glycolysis inhibitors affect glycolysis and oxphos? In particular, guessing how inhibitors that inhibit certain enzymes along the glycolysis pathway affect glycolysis is probably fairly intuitive. However, what's a little more up for debate is how do these enzymes inhibit oxidative phosphorylation? And because these 2 inhibitors affect different steps of glycolysis, and they have different mechanisms, do they show up differently when we monitor in-cell inhibition by Seahorse? So do these 2 inhibitors affect metabolism, cellular metabolism differently? And so the way that we're going to be testing this is by running the glycolytic function assay, which is a common assay used by people who work with Seahorse to understand how certain molecules affect glycolysis. So you might use this if you're saying, oh, hey, this molecule is a really promising glycolysis inhibitor. Let's see if it actually works. And so in this particular graph, we're looking at the ECAR, or the change in the extracellular acidification rate, and the changes in the milli-pH units per minute. But you'll also be looking at the OCR as well, or how the oxygen consumption rate, or by extension, oxidative phosphorylation, changes with time. And so there are 3 injections that you're going to be making in each experiment. And the first is glucose. So glucose stimulates glycolysis. All right, and so what happens when we add glucose, the extracellular acidification rate is going to increase a bit, And that's because you're bumping up the amount of glucose that the cells have, so they're going to increase their glycolysis rate accordingly to break down that glucose and to keep up with the supply. The next step is adding oligomycin, and oligomycin, it's an electron transport chain inhibitor. And so once we hit the cells with oligomycin, you notice that the ECAR is going to jump way up. And that's because as the cells are, as you shut down the cell's ability to undergo oxidative phosphorylation, they're going to compensate with glycolysis. And the next thing to ask is, okay, after if we add our glycolysis inhibitor, so 2-DG, 2-deoxyglucose and oxamate, what's going to happen? And that's what we're going to find out during this lab. You're going to predict and then see what happens, and then also see, do 2-DG and oxamate have differing effects, and do they affect the oxygen consumption rate differently? So again, here's another diagram that shows you that that 2-DG, again, is an inhibitor of hexokinase for that first step. LDH, of course, comes here at the end, and then oligomycin is inhibiting the electron transport chain. So before we begin, I want to really quick run over how the experiment is going to work in the first day in lab. So I will call you, or at the beginning of the lab, we'll get together, we'll review this one more time, and then we're going to go up to the Seahorse room where Manish, who works in the mitochondrial energetic bioenergetics core, will be running us through the experiment, and you'll get to see him wash the cells, prepare the chemicals for the injections, and then help set up the plates for the experiment. So before the assay even begins, so a day, 2 days before, Manish is going to take care of culturing and then plating the cells. And then the night before, I'm going to go ahead and I'm going to take the cartridge. So that's the plate that has the sensors and then the injection ports. I'm going to incubate it in a calibrant in a non-CO2 incubator at 37 degrees Celsius. So think about for a minute, why would we want a non-CO2 incubator? I'll ask you about that in class, but it might help to look to see if CO2 plays a role anywhere else in metabolism. You can look back in the figures earlier in this handout. And design assay, well, I did that many months ago, so that step, we don't have to worry so much. So the day of the assay, I'm going to go upstairs and I'm going to prepare the medium. So I take the medium, I add any extra supplements that we need, and then I adjust the pH. Then, after we're done with our assay, we'll run upstairs, we'll see Manish, and then with Manish, you're going to prepare the compounds that we need. So this would be the glucose, the oligomycin, and then the 2-DG and oxamate. Or I may have those prepared ahead of time. I'm not 100% sure. But then you'll dilute the compound, so it's the correct, right concentration when we put it into the cartridge, and then we'll load the cartridge. And this will actually be done after we've prepped the cells. So the cells have been growing in a different media overnight. That's not the Seahorse media. So at that point, Manish will show us how to pour off the extra media we don't want and then replace it with the special Seahorse media that is needed for the glycolysis assay. Then once we've taken the cells, we'll take the cartridge that is loaded, we'll put the pieces together, we'll put it into the Seahorse, and then we'll run the assay. We'll wait for an hour. That's when you'll do your extra credit presentations. And then we'll run upstairs. I'll pull the data off the Seahorse with my flash drive. I'll post the data to the website, the Google Classroom website, and then you'll analyze. And so really quick of just a rundown of what our plate is going to look like for the experiment. This is going to be our oxamate plate, and this is going to be our 2-DG plate, and we'll have one per Seahorse because we have 2 Seahorses. So the top and bottom plates are backgrounds. Okay, so we keep those blank so we get an idea of what the noise coming from the plate looks like. Then we're going to have our two plates. Okay, and so then we're going to have triplicates for each condition plus or minus inhibitor. So B through D on each plate are going to be plus inhibitor. So oximate for the left-hand plate and then 2DG for the right-hand plate. And then we're going to have minus inhibitor for E through G. So that's going to be cells with no inhibitor added. And so that way we'll have our background, and then for each plate we'll have our cells with and without inhibitor, so you can compare to what happens to the cells when you add inhibitor and when you don't add inhibitor, and then you can look for any differences with how the metabolism is affected based on whether or not you add oximate or 2DG. And the next few pages, we have some group work activities that we may work on in class, depending on how much time we have after your presentation. So have a great week. I look forward to seeing you for the Seahorse Lab. And if you have any questions, you know to shoot me an email. Have a good one.